Assalamu alaikum everyone. Information box ticket lifestyle. Biotechnology. Unit 3 Recombinant DNA Technology. First, we will talk about the introduction. Recombinant DNA technology is one of the few techniques that make conventional biotechnology into modern biotechnology. Paul Berg, Herbert Boyer, Annie Change, and Stanley Cohen are the team of scientists that made the first recombinant DNA molecule in 1973. The field of modern biotechnology started when recombinant human insulin produced by bacteria was first marketed in United States in 1982. The efforts leading up to this landmark event began in the early 1970s when research scientists developed protocols to construct new types of bacterial plasmid or vector by cutting out and pasting pieces of DNA together to create a new piece of DNA, recombinant DNA, that could be inserted into host bacterium such as E. coli. We have also observed that yeast can be made to produce vaccines such as hepatitis B, plants having special properties such as resistant to certain diseases, pests and herbicides, and plants with superior nutritive qualities can be generated very effectively. These excellent goals of genetic engineering were achieved because of the advent of recombinant DNA technology. Definition: simply defined, recombinant DNA technology is the art of cutting and pasting genes. This technique encompasses a number of methodologies or tools that enable us to construct new combination of DNA, recombinant DNA or rDNA in the laboratory for different purposes. The rDNA molecule thus constructed can be introduced into an appropriate host cell where it can be multiplied and generate many copies. This forms the basic concept of process known as gene cloning or DNA cloning. Steps in recombinant DNA technology. The following are the basic steps involved in the process of the rDNA technique for gene cloning. Number 1. Isolate DNA from organism, example extraction. Number 2. Cut DNA with restriction enzymes to generate DNA fragments of desired size. Number 3. Ligate or splice each piece of DNA into a cloning vector, an artificial DNA molecule capable of replicating in a host organism. Number 4. Transform recombinant DNA cloning vector plus DNA fragment into a host that will replicate and transfer copies to progeny. As you can see in this diagram, the process of recombinant DNA technology. Tools of recombinant DNA technology. Since the process of making a recombinant DNA requires the precise cutting and stitching of DNA molecule, it involves a number of molecular tools, the enzymes, to cut and modify the DNA. The major enzymes required for making of an rDNA molecule are the following. Number 1. Type 2 restriction endonucleases. Number 2. DNA ligase. Number 3. Alkaline phosphatase. Number 1. Restriction endonucleases. Restriction endonucleases are enzymes that are a natural part of bacterial defense system. These enzymes restrict the growth and multiplication of DNA of bacteriophages in bacteria, and they are called restriction endonucleases. There are 3 classes of restriction endonucleases. There are type 1, type 2, type 3, and each is characterized by slightly different mode of action on DNA. Type 2 restriction endonucleases are used in recombinant DNA techniques as they can recognize specific DNA sequences and cleave at a site that comes within that sequence. As you can see in this figure, the process of restriction endonucleases and what it does to the DNA. In 1968, a group of 3 scientists, H.O. Smith, K.W. Wilkow, and T.J. Kelly, isolated and characterized the first sequence-specific restriction endonucleases HEN2 from Haemophilus influenza bacteria. Since these restriction endonucleases are responsible for converting the DNA molecules into a specific number of fragments by cutting the molecule at certain specific sequences known as restriction site, these enzymes are known as molecular seizures. More than 3,000 restriction enzymes have been isolated from over 230 strains of bacteria since the initial discovery of HEN2. Nomenclature of restriction endonucleases. The restriction enzymes generally have names that reflect their origin. The first letter of the names come from the genus, the second and third letter comes from the species of the prokaryotic cell from which they were isolated. Fourth letter, if any, indicates the particular strains of organism. Don't forget to like this video, subscribe to my channel and press the bell icon button. Roman numerals indicate the order in which the enzymes were isolated from single strains of bacteria. Here is an example of restriction endonucleases. Nucleases are further described by the addition of prefix endo or exo to the name. The term endonucleases applies to the sequence-specific nucleases that break the phosphodiester bond somewhere in the interior of the molecule. Nucleases that function by hydrolyzing phosphodiester bond from the end of the molecules are called exonucleases, as you can see an example from this figure. Many type 2 restriction endonucleases recognize palindromic sequence. The palindromic sequence read the same on both the strains of DNA in 5' to 3' direction. These are usually 4 to 8 nucleotides in length, as you can see in the diagram below. Type 2 restriction enzymes cut DNA and produce two types of fragments. Some restriction enzymes such as HEN2 and SMA1 cut the DNA symmetrically on both strains at exactly the same nucleotide position, leaving blunt ends, whereas others produce fragments with sticky, overhanging or stranded ends, as you can see in this diagram below. DNA ligase. This enzyme is responsible for the formation of phosphodiester linkage between two adjacent nucleotides and thus joins two double-stranded DNA fragments. The phosphodiester linkage is formed between the 3' OH group hydroxyl group at the 3' carbon and 5' PO4 group of the pentose sugar of adjacent nucleotide of two different DNA fragments or NICs created in a double-stranded DNA molecule. In recombinant DNA experiments, DNA ligase is used to join two different DNA fragments, plasmid vector and the foreign DNA, that are annealed by the sticky ends. There are different types of ligase enzymes from different sources, but the most frequently used one is T4 DNA ligase produced by T4 Bacteriophage. Alkaline phosphatase. In recombinant DNA experiment, there is always the problem of self-ligation and the reformation of original plasmid or the vector that reduces the efficiency of the recombinant DNA formation. This can be prevented by using alkaline phosphatase. We know that the presence of 3' OH group and the 5' PO4 group is a prerequisite for the ligase enzyme to act. If any one group of this is absent, the DNA ligase cannot function. Alkaline phosphatase can remove the phosphate group from 5' end of the DNA molecule, resulting in a 5' OH group. Now the only chance of ligation is between the vector and the foreign DNA, resulting in a recombinant DNA molecule, as the insert DNA has the 5' PO4 group. This enzyme is isolated from some bacteria, bacterial alkylase phosphatase BAP or calf intestine alkaline phosphatase CAP. Vectors, the vehicle for cloning. Vectors act as a vehicle for carrying foreign DNA into a host cell for multiplication. Usually, small circular DNA molecules of bacterial origin are used as cloning vectors. A DNA molecule should possess the following essential characteristic to act as a cloning vector: origin of replication, selectable markers, multiple cloning site or polylinker, and small size. Types of cloning vectors. There are 6 different types of cloning vectors commonly used in recombinant DNA experiment. They are the following: Plasmid-Cloning Vector, Bacteriophage Cloning Vectors, Cosmid-cloning Vectors, Yeast Artificial Chromosomes, Bacterial Artificial Chromosomes, Animal Plant Vectors. Number 1. Plasmid-Cloning Vectors. Plasmid-cloning vectors are derived from bacterial plasmid and are the most widely used, versatile, and easily manipulated ones. BPR322 vector is an example of plasmid cloning vectors. This was the first widely used plasmid vector. It has a number of useful features. Origin of replication. It carries a fragment of the plasmid PMB1 that acts as an origin of replication. Size. It is relatively small at 4363 base pair. There is room in BPR322 for an insert of at least 6 kilobytes base pair. Copy number. Reasonably high copy number, approximately 15 copies per cell. Selectable markers. It carries 2 antibiotic resistance genes, ampicillin and tetracycline. Cloning site. It carries a number of unique restriction sites. Some of these are located in one of the antibiotic resistance genes, as you can see in this figure as well. Similarly, PUC series vectors are extensively used as cloning and expression vector. These vectors have different 3 important additional features compared to PBR322. High copy number. A mutation within the origin of replication produces 500 to 600 copies of the plasmid per cell. Blue-white screening. A synthetic polylinker. This is a piece of man-made DNA that contains several unique restriction sites. Number 2. Expression vectors. In most of the cases, the main purpose of recombinant DNA experiment and cloning is to sufficiently multiply and amplify the inserted DNA fragment. But before that, don't forget to subscribe to my channel. But sometimes the aim of the process will be to produce large quantities of recombinant protein encoded by the inserted gene. This can be accomplished by incorporating the necessary regulatory elements along with the genes in the vector. Such vectors are provided with signal necessary for initiation and termination of transcription, such as suitable promoters and terminator sequences, and signal for translation initiation, such as start codon and a ribosome binding site into the vector upstream to the multiple cloning site (MCS). These vectors are known as expression vectors. Vectors of the PUC series are examples of expression vectors. Number 3. Bacteriophage vectors. These are the engineered version of phage macrophage that infect Escherichia coli. This vector has a double-stranded linear DNA genome having a size of 48514 base pair, 48.5 kilobytes. Another 12 bases at the end of this DNA molecules are unpaired and complementary, and therefore are sticky or cohesive and known as cos-site cohesive end sites. These cohesive sites are very necessary for packaging the DNA into the viral particle during the lytic cycle, as you can see in the diagram below. The lambda phage vector is designed in such a way that the central region of the lambda chromosome linear is cut with a restriction enzyme and is replaced with foreign DNA digested with the same restriction enzyme. This recombinant DNA is packaged in phage heads to form viral particles. Phages with 37 to 52 kilobytes insert replicate by infecting Escherichia coli. Phages replicate using and produce large quantities of 37 to 52 kilobyte cloned DNA. Number 4. Cosmid-cloning vector. Cosmid-cloning vectors were constructed by certain features of plasmid and the cos site of lambda phase. A cosmid can be defined as a plasmid that contains a cos site from the lambda phase genome. The vector replicates as a plasmid as it contains a CO1E1 origin of replication and carries foreign DNA inserts up to 45 kb in size. The simplest cosmid vector has a CO1E1 origin of replication, selectable markers including the antibiotic resistance genes and the beta-galactosidase genes, and suitable polylinkous sites and lambda cos site. Number 5. Yeast artificial chromosome. Yeast artificial chromosome are the vectors that enable the cloning of foreign DNA fragments into yeast. These are used for cloning of very large DNA molecules in the range of 500 to 1000 kilobytes. These vectors are constructed with yeast telomere sequences at each end. Yeast telomere sequences and the yeast ARS autonomously replicating sequences for replication. In addition to this, there are restriction sites for DNA insertion and selectable markers such as amino acid dependence and antibiotic resistance on each arm. Bacterial artificial chromosome. Bacterial artificial chromosome are the cloning vector based on the extrachromosomal plasmid of Escherichia coli called F factor or fertility factor. These vectors enable the construction of artificial chromosome which can be cloned in Escherichia coli. This vector is useful for cloning DNA fragments up to 350 kilobytes but can be handled like regular bacterial plasmid vectors. Like any other vector, bacterial artificial chromosome contain ORC sequence derived from Escherichia coli plasmid F factor, multiple cloning sites having unique restriction sites and suitable remarkable markers. Both yeast artificial chromosome and bacterial artificial chromosome were very useful in genome sequencing projects such as Human Genome Project. Animal and plant vectors, Shuttle vectors. There are some vectors developed for transforming plant and animal cells. Numerous vector systems have been developed for use in eukaryotic systems. The most common of these are often called Shuttle vectors as they replicate in both prokaryotic and eukaryotic hosts. In general, DNA manipulation and characterization are done in prokaryotic systems and then manipulated DNA is reintroduced to eukaryotic system for functional analysis. The common features of such Shuttle vectors are following. They are capable of replicating in two or more types of hosts including prokaryotic and eukaryotic cells. They replicate autonomously or integrate into the host genome and replicate when the host multiplies. These vectors are commonly used for transporting genes from one organism to another, that is, transforming animal and plant cells. Mammalian vector. These are Shuttle vectors developed for use in mammalian tissue culture. These eukaryotic origins of replication are typically derived from well-characterized mammalian viruses such as simian virus 40 SV40 with SV40. SV40 was the vector used in the first cloning experiment in mammalian cell culture in 1979. Since there are a number of mammalian vectors based on viruses have been developed to transfer genes in mammals for various purposes including gene therapy. At present, retrovirus-based vectors are the most commonly used vectors for the cloning genes in mammalian cells. In addition to the origin of replication, these Shuttle vectors always carry antibiotic resistance genes which function in eukaryotic cells, that is neomycin resistance and hygromycin resistance, etc. Plant vectors. There are a number of Shuttle vectors developed for the genetic transformation of plant systems. The most successful Shuttle vectors developed for the plant system are those that are based on Ti plasmid of Agrobacterium tumefaciens, a bacterium that causes tumor formation in plants. This has been successfully exploited for the development of Agrobacterium tumefaciens based vector for the genetic engineering of plants. Here is an example of the Agrobacterium tumefaciens vector. Host cells. The ultimate aim of development of recombinant DNA molecule or vector is the multiplication of rDNA by cloning in a suitable host cell. There are many types of host cell available for the purpose of cloning. The host cells can be bacteria, yeast, plant, or animal cells. Making recombinant DNA. The first step in the construction of a recombinant DNA is the isolation and purification of vectors and the DNA fragments containing the genes to be cloned. First, digest the vector DNA with a suitable restriction Endonucleases and make it linear with or without sticky ends. Next, isolate the DNA fragments carrying the genes by digesting the genome or the source DNA strands with the same restriction Endonucleases used for cutting the vector. The linearized digested vector and the target DNA cut with the same restriction enzymes are incubated together in the presence of DNA ligase. During incubation, the sticky ends of the two DNA strands come together by base complementation and become hydrogen bonded to each other. At this point, a phosphodiester linkage is established between them, mediated by the DNA ligase enzyme. This results in the formation of recombinant DNA molecule of vector and the target DNA. As you can see in this figure, the making of recombinant DNA. The disadvantage of this strategy is that there is possibility of re-annealing of the sticky ends of the linearized vector, leading to the formation of vector itself without any foreign DNA fragment. This problem can be reduced considerably by adopting any one of the following strategies. Number 1. Treating the digested vector with alkaline phosphatase to remove the 5' phosphate group. Number 2. Restriction digestion of the vector and the isolation of foreign DNA fragment with 2 restriction enzymes instead of 1, double digestion, so that the 2 sticky ends of the vector will not be identical, which will prevent the re-annealing of some extent. Transgenic introduction of recombinant DNA into cells. After the construction of the recombinant DNA molecule, it has to be introduced into the suitable host cell so that the DNA will multiply. The following are common methods used for introducing the recombinant DNA molecule into the host cell. Number 1. Transformation. Number 2. Transfection. Number 3. Electroporation. Number 4. Microinjection. Number 5. The ballistic method. Number 6. Lipofiction. As you can see with the diagram, transformation is the most commonly adopted method for introducing a recombinant DNA into a host cell. During the process of transformation, cells take up foreign DNA from their environment. But many cells, including Escherichia coli, yeast, and mammalian cells, are not able to take up the DNA from their environment naturally. Certain chemical treatments can enhance the ability of cells to take up the foreign DNA or make the cell competent for transformation. For example, Escherichia coli cells become more competent when these are incubated in cold calcium chloride solution. Transfection. Transfection is another method used for the transformation of cultured cells. In this method, DNA recombinant vector is mixed with charged substances such as calcium phosphate, cationic liposomes, or dextran and overlaid on the host cells. This ultimately results in the uptake of the external DNA by these host cells. Electroporation. During the process of electroporation, an electric current is used to create translucent microscopic pores in the cell membrane of the host cell, as you can see in the diagram. Through these temporary openings, foreign DNA enters the cell. This method is especially suitable for cells such as yeast, mammalian cells, and plant protoplasts. Microinjection. Microinjection is a specialized technique by which the DNA fragment or genes can be directly injected into the nucleus of plant and animal cells. The procedure involves the direct injection of the DNA into the nucleus of the host cell with the help of a glass microinjection tube or syringe. The instrumentation for a microinjection consists of a microscope of high magnification attached with fine needles, syringes, and other facilities to carry out delivery of DNA into the selected cells automatically. Ballistic method. The ballistic method was developed for the introducing foreign DNA into plant cells with the help of a gene or particle gun. During the ballistic method, microscopic particles of gold or stagnant coated with the DNA of interest is bombarded into the cells at a high velocity. The bombardment of the particle is carried out with the help of a mechanical device called a gene gun. Lipofixation. It is used to transform all cell types. DNA to be transferred is placed into liposomes, which are small lipid vesicles. The liposomes fuse with the part of the cell membrane of the host cell and the contents. The new DNA enters the cell. Targeted DNA encapsulated in a spherical lipid bilayer termed a liposome. In the presence of PEG, endocytosis occurs. After endocytosis, the DNA is free to recombine and integrate with the host genome, as you can see in this figure. Identification of recombinants. During the process of genetic transformation, a very low percentage of the total cell population takes the recombinant DNA. Therefore, it is very important to have an efficient screening method to select the transformed cells. There are various screening techniques that are based on the expression or non-expression of some of the traits present in the vector along with the cloned genes. Some of these traits are the resistance to certain antibiotics. If the antibiotic resistant gene is present along with the cloned gene, it is very easy to select the recombinant transformants directly on a medium supplemented with respective antibiotic. In most of the cases, there are two stages of selection. Number 1, selection of transformed cells, that is the cells that have taken a plasmid. Number 2, identify the transformed cells that have the recombinant plasmid. Stage 1, the transformed cells containing a plasmid and can be identified by positive selection method or negative selection method. The most common markers used for positive selections are the antibiotic resistant genes present in the cloning vectors. By plating the transformation products directly on antibiotic plates, all untransformed bacteria die and only containing those plasmid with the antibiotic resistant marker genes can grow to form colonies. By this selection method, we will be able to get the transformed cells with the plasmids. But it is not possible to identify the transformed cells or colonies having recombinant plasmid from the transformed cells or colonies with parent plasmid without any insert DNA fragment non-recombinant vector. Stage 2. A negative selection method can be adopted for identifying the presence of recombinant vector among the transformed cells. In order to identify those plasmid carrying a foreign DNA fragment, the multiple cloning site in the vector is engineered in such a way that the insertion of a foreign DNA strain disrupts the expression of a selectable marker genes, a phenomenon known as insertional inactivation. Two types of selectable markers are used for negative selection. Number 1, the insertion inactivation of antibiotic resistance. Number 2, insertion activation of an enzymatic activity. A vector carries both an ampicillin and a tetracycline resistant genes. The phenotype of bacteria containing the intact plasmid is ampicillin resistant and tetracycline resistant. Insertion of foreign DNA into the PEST1 site located in the ampicillin resistant gene results in an ampicillin sensitive and tetracycline resistant phenotype. Conversely, insertion of foreign DNA into the Escherichia coli RI-HIN3 or SAI1 site located in the tetracycline-resistant genes results in an ampicillin-resistant and tetracycline-sensitive phenotype. Here is a diagram of step-by-step procedure. Insertion inactivation of enzymatic activity. This system is based on the insertional inactivation of LAC-Z gene. The LAC-Z gene is a component of the LAC operon and it produces an enzyme beta-galactosidase. This enzyme can cleave a colorless synthetic substrate X-Gal into a blue-colored product. If the beta-galactosidase gene, LAC-Z gene, is disrupted and inactivated by inserting the foreign DNA fragment into it, X-Gal will not be cleaved and the development of the blue color will be prevented. Thus, the selection media is provided with the Gal-X and the host cells that carry normal vector without insert DNA will produce blue color clonies, and those cells which carry the recombinant plasmid will develop white color clonies. Here the positive selection expression of antibiotic resistance and the negative selection inactivation of an enzyme activity beta galactosidase can be combined together and carried out in a single experiment. Here is the diagram of screening blue and white selection with step-by-step procedure. And that's it for today. Thank you for watching. Don't forget to subscribe to my channel for more videos. Thank you.